Good morning, everyone. Thank you to Gabriel for inviting me. Today, as I said, we're going to talk about a bit about the omic techniques to study microalgae. A little bit about myself. I come from the group ParAqua-COST. I started as a molecular biologist, and then I went on to do studies on nitrogen pollution on intensive farms from biological point of view, chemical and isotopic analysis, and gaseous emissions. And then I went back to the molecular biology, so I did some study on quorum sensing, so how bacteria communicate to generally on pathogen of rice. And then I went on to the work in University of Bolzano in Milano, studying, characterizing the microbial communities of, for example, wastewater treatment plants and soil communities. And currently I am covering a position as a permanent microbiologist at the Italian National Agency for New Technology, Energy and Sustainable Economy. And I'm also doing a research tenure track at the University of Milan, always with the group ParAqua-COST, where I'm mainly studying, for example, communities of microbial fuel cells, how to use bacteria to improve the yields of fertilizers, and of course soil communities, and sometimes microalgae. The group ParAqua-COST is located in Milan, in the heart of the agricultural place of Italy. We work mainly on three fields, so environmental protection, agriculture, and bioeconomy. So all the thematics about soil, restoration of soil, soil bioremediation, and up to the use of digestate, LCA analysis, biogas, biotransformation, and green chemistry. So today I wanted to talk about a bit, starting with the microbiome in general, then going on to what are the omic approaches that we can use to study the microbiome. Then I will do a short excursus on the type of next generation sequencing that there are, and then a couple of cases of study. Oh, but this is an old presentation.
No?
Yes.
I paste your presentation in this folder.
Wait one second.
Okay. Okay. 1 second, because we will try to modify the presentation. Ahora se escucha mejor. Please be closer to the microphone.
Yes.
Yes.
No.
This is the... Yes. Try again.
Okay.
Okay.
Let's go with the PowerPoint, the PDF. Can we do it?
Yes, yes, no problem. Okay, yes, because you are already sharing the
Okay.
screen.
It's okay like this, huh?
Yes. I think there is an option for full mode, but I don't know exactly. That's
Okay. And then, yes, and then this afternoon, I'm going to walk you through a workflow for the NGS analysis. So in theory, following all these following all the steps, you will be able to do NGS analysis from DNA extraction until data analysis. So what is the microbiome? The microbiome is defined as a characteristic microbial community that inhabits a well-defined habitat with physiochemical properties. The microbiome is composed mainly of the microbiota, which is the community of bacteria, archaea, fungi, protists, and microalgae, which lives in a commensal, symbiotic, or pathogenic type of way, so they have positive or negative interactions. However, when we talk about microbiome, we also speak about the theater of activity, so all the environmental condition, all the molecular that are produced by this microbiota. So if you want to study, we need to study the whole microbiome, not just focusing on the microbiota. What is also important is to study the microbiome, of course, is always evolving. So it's important to study as a dynamic and interactive microecosystem. So when planning an analysis, you should always plan to have repetition in time and scale when talking about microbiome. And what is important also is that generally the microbiota has two typical composition of species. So one is the core, which generally tends to remain across all perturbation that can happen, but also a transient microbiota, which is always changing and can be always new. So we need to consider all these parts when analyzing a community. The main approaches to study a microbiome are the traditional, which is culturomics. So you take a sample, you isolate it, and you study the single isolated microorganism. The advantage of these traditional approaches is that you can study a bacteria in its dynamic and changing changing conditions, so across a time scale. And also is development across, for example, when changing the parameters, also changing environmental variables. However, the problem is that only a very small amount of bacteria and microorganisms are culturable. So now they are speaking about, it was 99% that are unculturable, now it's a bit higher because there are new techniques to improve medias and to target the media on the genome of the plant, of the microorganism. The new techniques are based on metagenomics. So you take your sample, you extract the DNA, you go through the sequencing, and then you analyze the genes that are inside this microbiome, and also the RNA, so the function of this microbiome. The advantage of this is that you can consider a higher amount of microorganism, also the uncultured one. Main problematics is that you don't know if they are viable or not, and it's only a snapshot. So it's a picture of this community at a specific time in a specific place. I wanted to point out that maybe for now there is a new approach, so the reverse genomic approach, which enable to predict the functional metabolic pathways of a community, which is now being used for create ad hoc media. So, for example, for microalgae can be interesting because you can target a specific microalgae with specific media to grow. What are the types of omic approaches that are present at the moment? So starting from the central dogma of molecular biology, we aim to target DNA, RNA, and protein. So you have metagenomics that can cover covers the study of genetic material directly for environmental samples, and you can target specific organism with amplicon sequencing or the whole community with the whole metagenome approach. We have the transcriptomic, which is the pathway is similar to the metagenomics. However, it works on generally mRNA. So you are able to know which genes are actively transcribed. So what is the community doing at that point in terms of transcription. And then you have all the proteomics, which target the proteins, so the enzyme present at the moment. And now you have also metabolomics that is recently have been included in the definition of the techniques used to study the microbiome, because also the metabolite are important, for example, to quorum sensing and microbial community, and can shape a community. A little bit about the sequencing, the history of sequencing. So it started in 1987 with the Sanger sequencing, which is the classical approach. Then it went on to 2005, where we have the next generation sequencing, or sequencing by synthesis, which brought high throughput, so massive parallel sequencing. And one of the main leaders in this type of sequencing is the Illumina, which is also already still currently used. And then you have, in 2008, the third generation sequencing, which work on single molecule DNA. So it enables, with the high throughput of the Illumina, it allows to have longer reads, so a longer strain of sequencing. How do we select this specific, one of the specific methods? It depends on your research objectives. So it depends on also you have the isolating strains or the whole community. For example, the Sanger sequencing is very useful if you have a low amount of data of samples. And it's very simple, the analysis, you just do blast and you have your identification. However, the low throughput is a problem. The Illumina is currently the one with the highest sensitivity. So if you want to detect a mutation, this is the way to go. However, for a small amount of samples is at high costs, and the lengths are very short. On the other hand, the nanopore technology is a longer approach. It's interesting because it gives you a direct sequencing of RNA. You have longer sequencing, but the costs are higher, and the error rate is very high. It's very high. It's higher than the Illumina sequencing. How the Sanger sequencing works? So you have your DNA. You extract your DNA. You select the region that you want to amplify. You amplify, so you have multiple copies of your fragment. Then you create 4 solutions with dideoxynucleotides, and you start replication. With this replication, you will have different type of segment, a different type of length, because once the deoxynucleotide is included in the replication, the polymerase stops. And then with the electrophoresis, you are able to obtain these that you see in the corner below, this electropherogram, which gives you the all the reads at all the bases at each positions. This system can work, can sequence up to 1,000 base pair, and it's mainly used now to, for example, if you have isolated samples, you amplify your region, and it gives you what bacteria you have or what microorganism you have, and nothing more. High accuracy, low throughput, as I say. But this is where we start. And if you want to use this on your samples, pretty much with 20 euros, you can identify an organism. Then we have the next generation sequencing, so sequencing by synthesis, which we'll talk about the next slide. And is not very feasible to have in your lab if you're not working a lot on this, but you can work. There are some different type of machine, from bench top scale to production scale. Of course, once you go higher, the cost increase, but the cost of the machinery increase, but the cost of analysis decrease. But generally is feasible only for sequencing platforms. And then we have the long read sequencing, which is very interesting because how it works. It works like you have a pore on a membrane. You have some proteins that take your copy of DNA and brings it to this pore. The single chain of DNA goes inside this pore, and it change as it passes through each bases change the potential of the membrane. So each base has a specific change of potential, so you can have all the sequencing for each base. At each change of potential, you have a single base. And as you can see on the right, the dimension is very small, so the machine is like 10 per 3 centimeters, so everyone can have it in the lab in a drawer. How do you organize an NGS workflow? So first, you have to plan the study and do the realization. Then you collect the sample. Be careful to be as sterile as possible, depending on the condition. Of course, if you work with wastewater, you don't need to be that sterile. Then you do the DNA extraction. And then you do the sequencing, then you have all the major problems with the informatic management of the data because it requires a lot of calculation potential, and then also space in a server. And then you go on with the statistic once you have the table of taxonomy, and then you go on to do your study. This slide is also is only to say that there are some parameters to consider before you start. The main ones are the sample sizes and the replicates. Generally, when working on microalgae from an engineering point of view, you tend to do only one sequencing, which can be dangerous because depending on the hand of the worker, if he's not very experienced, the result can change greatly. So at least if you cannot have a biological approach replicate, at least to have some technical replicates, which generally I never see in the engineering studies. They are just descriptive, but if you only do one replicates, then the statistic always fails. And then you can also include some type of control, positive control, negative control, some synthetic communities that just tell you all that your process is correct. And then in this afternoon, we'll speak about also how to extract DNA, because sometimes if you work, for example, in microalgae on wastewater or grown on digestate, you can send this sample to an external platform. It will cost you 40 euros, but for example, for these matrices, it's better if you learn to do it yourself because most of them won't do it. And also you can, knowing your matrix, you can create an ad hoc extraction, which will give you better results. So for the microbial genome studies, there are two main ways. As I said before, you can start with the isolated culture. So with an isolated culture, you can have two types of sequencing. So you can work on the resequencing of the whole genome. If you have it's already sequenced bacteria or microalgae, you can do the resequencing and compare if your sample is the same or if it has variations in the DNA, genomic DNA. Or you can do a de novo assembly. If you don't know your algae or whatever, you can start and do a complete sequencing from the beginning and characterize the whole genome. Then there is another option, so working on the total DNA. On the total DNA, you can have two types of ways. So the amplification of a specific region with amplicon sequencing, which target a specific part of the ribosomal RNA genes. Or you can go with a shotgun genomics, which gives you the whole community. It extracts everything and also all the functional genes. Talking about the amplicon sequencing, which region do you select? Generally, it works on the ribosomal. For the bacteria, is 16S. For the fungi, is the ITS, which is always a ribosomal gene. Or for microalgae, you use the 18S. Why you choose this sequencing? The sequences, because these are regions that alternates very conserved part but also a very variable region. Why? Because the ribosomal RNA is it's used for producing the protein, so it's a very essential function in the cells. So the function needs to be conserved. So, for example, the region where you have the enzymatic site are very conserved across all bacteria, across all microalgae, and so on. But the region that are not extremely functional to the enzyme can variate, and these are very variable, hyperly variable. So what you do is you create the primers, which are required for the amplification on the non-variable region, and you try to amplify the very variable region in the middle. So with only one primer, you can amplify multiple, they're called universal primer. You can amplify multiple species and with one shot. Yes. What gives you the amplicon compared to the shotgun is more sensitive because you are enriching your community with PCR. So you're targeting a specific region and you have multiple copies. So even if you have a low copy and maybe the amplicon won't get it, amplifying you have higher probability. However, what is the problem is that you have a PCR bias. So you have to know what to amplify. So it's based on the selection of the primer, which can be good, or for example, if you don't know, if you don't target, if you want, for example, if you want to target a specific fungal community, maybe because the primer of the fungi are have been until now produced on the soil microbial community. If you're trying to work on pathogens, for example, fungal pathogens, your universal primer might miss, so you want to go and select a new region, which you will not see with the universal ones. On the other side, we have the shotgun. So you just take the whole DNA, you prepare it for sequencing, and you just sequencing. This has a lower bias because you're amplifying pretty much everything and is independent to your prior knowledge. But of course, it's a higher amount of data and more complicated to analyze. How the shotgun sequencing works, it has 4 main steps. So the preparation of the DNA, then you are amplifying your specific region, your fragments, then you go on to the sequencing, and then you go and align the data and have your taxonomy identification. So the sample preparation, it works. You extract your DNA, you fragment it, then you just clean it. So you can have you need to have blunt ends. So through enzymatic reaction, you clean these ends. Then you add the tailing, which is the addition of adenine at one of the ends, at the end generally. And then you adapt a ligator, which can work for the helps you to ligate the fragment to the sequencing platform. And then you have the size selection, because for Illumina sequencing, you can only have up to 500 base pairs, so you need to select this type of fragments. Once you do this, you will have this type of structure. So you have your DNA inserted, which is your target. Then you have primers to start the amplification in the next round. Then you have the indexes, because you can have multi-parallel sequencing. So you need then a tag to say what sample is it. And then you have the adapter, which help you with the sequencing. So as you see here, you see how it works, the amplification. So you have your primers, you have the primers with your adapter, then thanks to the adapter, you attach it to the flow cell, which contains millions of oligomers where you can attach your adapter. Then the next step is the bridge amplification, where you start one filament of DNA attached to each of oligomers. And so you can amplify by region your target sequence. and you will have a cluster formation. You do this one time for the forward strains, forward read, and one time for the reverse read, and you will have the formation of clusters, which all are copies of your original fragment. So you amplify the option to have a representation of your sequencing, because that sequence will be present only in 2 copies. So this is amplifying the signal. And then you have the sequencing start. So it's sequencing by synthesis. So you have a primer that attached to your primer region below the adapter. And each time one base is attached from the polymerase, there is a fluorescent signal that tells you what base has been attached. And this is parallel to all the flow cells, so millions of light signal at each moment. And every round you will have one more base, which is then detected from the scanner, let's say. And then you will have your all your reads. What is the output for the sequencing? So there is a FASTA file. You have the first line is your sequence identifier, so you know what sample is it and what sequence is it. Then you have the sequence, which is just of course, the repetition of your basis. Then you have a spacer, and then you have your quality score. So this quality score tells you if your sequencing was good or not. And then you have, there is a specific tool to use, the FastQC tool, which you can use to analyze this quality score and to visualize it. So you can see here in the corner, it gives you the length of the sequencing, it gives you the GC content, and you can see, for example, this is a sequence. You can see that, for example, the first part of the sequencing is good, then the quality tends to drop, and because generally the sequencing works, the results generally are always bad at the end, because your sequence is for a long time, you're reaching to the end of the chain. So you always see this kind of worsening of the fragment, and also the reverse primers are always much worse, because it's the second round of sequencing, so the quality is bad. And you have to account this when you are going to do the analysis later. How are you going to analyze these results? First, you need to quality check and clean all your data. So this can be done if you are using an external provider. This can be generally done by them. So you need to remove all the adapters, the primer sequencing. You have to do a quality control, and you need to separate the forward reads, reverse read, and each sample. If not, there are some programs like Chime that you can do this for you. Then what do? You select, there are different tools. For example, there is Chime, but you need some coding abilities. And then the main one are Chime and Dada2. Dada2, it's easier, and we will see it later, because you can use it on R, so you don't require to know Linux and how to code. But what all these workflows do is just you clean, you check for chimera, sorry, which are fake sequencing. You eliminate the bad sequencing, and then they give you the table to then analyze statistically. What is the main difference between Chime and Dada2? So one works on operational taxonomic unity, the other one on amplicon sequence variance. So you can see in the first picture what the samples that you have, the sequencing. For example, in your sample, you have 4 types of bacteria, so 4 types of sequencing, 4 types of sequences. Then you go on to the amplicon, do the amplicon analysis. What happens is that you introduce errors, so your sequencing can become more sequences. So for example, you see the red, you have one sequencing, but with the error, it becomes 8 different different strains, reads. So what do you do? The OTUs, so CHIME2 gives you practically it clusterizes everything at a 97% similarity. So the problem with the OTUs, which are now less used always, is that, for example, you lose some data. So, for example, those two different microorganisms are clustered together in one. What does error 2 and AWS analysis instead is to learn an error. It does an error. It has algorithm that check the error, and it tries to go back to the original sequencing. So it infers it from quality scores and resolve the problem, understanding if the sequences are real or not. Another point in favor to Data2 is that it can differentiate sequencing by one nucleotide and has a higher capacity to recognize fake sequences, so chimera, and it will gives you the same AWS. While if you want to compare different studies, the OTUs, you will have to reanalyze everything again. While the Data2, you can just combine the table and you're done because the AWS are always the same, while the OTU change with every data you input. Then this is what we're going to see also better later, but for the one online, how it works Data2. So after the pre-processing, which cut the primers, the multiplex, so you know what samples are, you know the samples, and you have the trimming and filtering. So after this process, you have the taxonomic assignation. So you do, it gives you from, it makes you, like the output from Data2 will be like a taxonomic assignment. So you will have your samples, your AWS, and then the abundance of each of your sequence. And will also give you all the taxonomic levels, so from phyla to species. You can use different database to do this assignation. The main ones are Silva for bacteria, which is always which is always continuously updated. For fungal and fungal-like species, you have the Unite, which is the main one to be used. For microalgae is a bit of a problem because none of these are actually very comprehensive of microalgae. So what you can do, you can try with the ITS and 18S database, but what you can do is also go directly. There is a tool for R, which I will explain this afternoon, that you can directly go to BLAST and take from there the sequencing. So it will always be updated. After you have your data set, then you can have you can go on to do multiple analysis. The main one, which also if you send your samples to a server asking for also the bioinformatics, will be evaluation of the diversity, the richness, and then pretty much the biomarkers. So the taxonomy will come out something like this. You have all your steps of all of your taxonomic groups. One main problem with bacteria is generally they are very similar. So your identification will stop maybe at the genus level. But for microalgae and fungi, generally you always reach the species level. What can give you a richness and evenness analysis? So once you have your data, you can go on to do this analysis. The richness gives you just the number of species in a sample. You can have also the Chow index, which accounts for all lower abundance species, but generally an observed richness is okay. Then you go to on to analyze the relative abundance. So the Data2 will give you the number of species accounting from the total number of species. And you can also have the analysis for the evenness. So as you can see in the picture below, they even tell you how fortunate are each species within a sample. So for example, if they're 50-50, as in this first case, you will have a higher evenness. If, for example, one species has only 2 components while the other have 5, you have a lower evenness. So it gives you an analysis of this type of data. One is more interesting, for example, also for microalgae maybe, is alpha and beta diversity. What is the difference? The alpha diversity gives you the diversity within the sample. So how many unique species, how many shared species. And you can have generally 2 types of diversity. So Shannon is very used, and it gives you higher stress on rare communities, or Simpson, which works more on evenness. So how are your the dominant stuff, the species. And also you can use beta diversity, which gives you, instead of the of a single sample, gives you the relationships between samples. So it estimates how much your community varies. So you can do this also for different type of reactors. You can have an identification of the diversity among them. Then you can work on biomarker identification. So for example, you have the LEFSE tool, which can be used also as a platform, online platform. What does it give you for each samples? It tells you what species are enriched in the sample. So mainly also what species are driving your communities. And it gives you also the similarity between them. For Amplicon, even if it's not a shotgun, you can do an analysis which can tell you the potential of your community. So it's not, it gives you like what is the maximum of the maximum functional properties that I can have. There are several tools, both for R, both for not R, or depending on if you're using OTU or AWS. But as you can say, you can select a specific enzymatic pathway, and it can tell you, depending on the community, from what bacteria or microorganism you have. It gives you, okay, you can have a total amount of, for example, this specific gene for nitrogen fixation. You can have this, and this is the maximum you can have based on your population. And it gives you also all the pathways connected to all the metabolic pathways. If you are not, if you want to dive further in the functional notation and gene distribution, you have to go for the shotgun. As you can see from the first picture, the workflow is pretty much the same. So quality check, taxonomy annotation, and then you go and use a specific database for functional annotation, and can give you more a picture of your real enzymatic properties of your community. And it can tell you what are the, for example, the genes that contributes to this enzymatic file. However, this is based also always on DNA. So if you want to go what is really happening in this community, you just go with the transcriptomics. Also here, you have different types of transcriptomic analysis, which can go from the metatranscriptome, so the gene expression of the whole community, or you can target specific microorganisms for prokaryotic. You can have work only on those, or also on eukaryotics, or for example, plant RNA. And it gives you also, if you have a control or different type of experiment, it gives you the differential expression. So what is the difference between them in terms of functional profile? Now a bit of the real life. So this is I took a case from a project we did with the University of Almeria and Gabriel's group. So within Sabana, Sabana aimed to develop a large-scale reactor based on using, for example, a low-cost a low-cost media for water, for example, for growing microalgae, for example, sewage, centrate, and to compare it to also to synthetic fertilizer. So we did several studies on microbial communities in this European project. And for example, I take this one. In this study, we aim to differentiate the community between two different types of reactors, so between cascade reactor and raceway ponds. The algae were grown on wastewater. What we did was we collect the different type of algae so it was chlorella, Schnedesmus, then chlorella plus Schnedesmus, and Nostoc piscinalis. So we went to collect samples for all these reactors, and then we did an analysis of the community, so characterization, and then tested the resistance of multiple microalgae, so the inoculo in this system, and also to understand what positive correlation can we have within the microbial consortia. So what we did was we extracted DNA. How we extracted it, we used lyophilization to concentrate the samples. Then we went to do a 96 well extraction. We did the analysis, an amplicon analysis to target bacteria and the cyanobacteria, and a fitness analysis to target the eukaryotic microalgae. And we used the Illumina sequencing. This study then was analyzed using OTUs. And then we went on to use some statistical analysis, such as the one I just showed you. So observed richness, diversity, beta diversity, and the occurrence and interkingdom relationships. This was all done using R packages. What we achieved, for example, here you have the table of the beta diversity. Then you have below you have the bar plot, which tells you what are the most abundant species. So I think this one was above 3%. And then the NDS plot is a graphical representation of the beta diversity. The first bar plot tells you what are the microalgae, and you can see that the orange one is the chlorella. So chlorella took pretty much all the reactors, so there was a contamination by chlorella, which proves to be unfortunately the most robust strain and almost an invasive species. With the beta diversity, we compared the effect of the TLC and the raceway, and it seems that the TLC compared to the raceway did not favor eukaryotic microalgae and supported higher variability. So we saw that the richness and the diversity was higher in the TLC. While when looking at the prokaryotic community, we saw that the Nostoc piscinalis was maintained even when we had the chlorella in the reactor. And we saw that for prokaryotic, the community was much more diverse between the two reactors than the eukaryotic. So the TLC lead to higher variation of our community. Then what we did was a potential study of the functionality. We went on to look at the nitrogen cycle, and we saw, for example, you have here a heat map that tells you which genes. So on the left, you have all the genes involved for nitrification, denitrification, the RNA. And this told us that, for example, the TLC, sorry, had a higher potential of carry out nitrification, and while the RV higher the nitrification, which was also proved from the chemical point of view in this experiment. Then we went on and do a co-occurrence analysis. So this analysis tells us, as you see below, what are the negative and positive interactions. So positive are the red ones, the significant positive interaction, and the negative are the blue ones. For example, we saw that with chlorella, which is the highlighted in red, you have a higher amount of interaction, negative with, as expected, with microorganism that feeds on algae. So, for example, we had also protists, I think, and other pathogens. While, for example, we saw that microalgae, we were able to select bacteria that were having a positive influence. So probably they are using it. They have like a growth promoting factor to them. So with this, we were able to identify those. Then very quickly, a study on shotgun. This was done in collaboration with the University of Trento in Italy. They wanted to see the difference between a photobioreactor, a column photobioreactor, and a traditional activated glass system to treat wastewater. Similarly, we went to do an extraction. This time we use a centrifugation for reduce the volume of the microalgae because the percentage of water was much higher, so lyophilization would take a very long time. Then we went on to use the Illumina sequencing with pair and shotgun. We use different type of bioinformatic analysis. So we use the standard, which is Clagnan 2 and Bracken 2 for the taxonomic assignment. And then for this, you need to do some coding. But then we went on to carry out the functional contribution, and we used a online platform to do this analysis. And then we also used a platform to carry out a network analysis. And then the statistical analysis are pretty much always the same. Also here we saw a higher profile, we saw a higher abundance of gene related to the pathogenic due to, of course, the nature of the media. We also here, we went to check the nitrogen cycle genes. The difference here is that this is not potential, so this is the actual structure of the community. And we saw that, for example, even if you have the two, with the beta diversity, we saw that even if you have two very different systems, the biomass is different, but the effluent is more similar to between them. And what is interesting here is also that we went to look for pathogenic potential. So we went to look for each pathogen, and we were able to see the from the wastewater, so the inlet to the outlet, we saw the changing in the community of pathogens, and we were able to see that microalgae had like a disinfectant effect, but depending on which type of pathogen you are analyzing. And here you can see also with this analysis, we're able to go into deep in all the energetic pathways. So you can have carbon fixation, methane metabolism, and go on, and you can go deeper to the enzymatic level. And also here we found that, for example, the microalgae bacterial flux enhanced the nitrification when compared to the conventional sludge system. Here is an example of what you can achieve with the co-occurrence analysis. So also here we were able to see positive interaction with bacteria which stimulate plant properties, but this is much more in deep analysis when you are using the amplicon sequencing entity.
Thank you, Elisa, for the excellent presentation. It's really huge amount of information. Please, I don't know if do you have some questions from the audience? Also, I think that it's possible for the people attending online using Zoom to activate their microphone and to provide questions if they want to do that. No questions? Yeah, still you are. Yes, Beren, please.
Yes.
But still most of the studies
Allora, they used to use Otus. Now pretty much everyone is changing to, well, now in the last couple of years, they are using more as SU because they give you more clear results and more realistic. Even if it removes a lot of data because the quality check is very strong, the one that you have are more one sequence, one result. While in the OTUs, you have a cluster, so it's general. It's not very specific. And you always you cannot confront the data between studies, while with AWS, one AWS, it will always be the same across all analysis.
Elisa,
I think it will be easier with the workflow later.
okay. Only one remark. The training activity will be not performed this afternoon. Will be performed now after the coffee break. Okay? No problem. One question from my side. I understand that this first question, this type of methodologies are improving a lot every day. It's incredible. I never heard about the nanopore technology, etc. It's so small, it's so practical. Yes, it's
Like this,
really.
yes.
But
you also have the scale up, which are not big, but bigger.
Yes. In my opinion, the most important fact in this technology, or the most relevant factor, is the expertise, being able to manage this technology, etc. I understand, and I think that for research purposes in the university or whatever, experts like you are the most suitable to be able to perform this type of analysis, whatever the methodology, etc. But how do you think that the companies can implement these methodologies in their facilities?
Well, I think if they have the resources to do it, they can create a small part of their lab to, like you train one person and they can do it. Otherwise, you go to for external sequencing, but it requires, as we say last time, time and high resources. No, high resources not, but time. So you don't have an immediate effect of your data. So if you want to use it as a control or what is wrong, I want to change something, it will take some time. So it's not immediate, the result.
Not immediately, but can you imagine that certain methodologies, probably the simple ones, can be used regularly in production sites to evaluate or to certificate the absence of contaminations? That you are producing a pure strain, is always the same strain or whatever, or including this case is not suitable?
This afternoon we will see a study we did with also Almeria. We compared the different technologies for pathogen identification. So, for example, this study told us that a lot of pathogens are overlooked, but you can get it with the sequencing analysis. However, the sequencing doesn't tell you if they're alive or not. So if you want to do like a preventive, maybe it's useful because it tells you, okay, you can have this, maybe be careful in this part of your process. But if you want to know, like if you want to know if they're alive, the only way is, I would say, culturing. But of course, culturing, you cannot cultivate everything. Now there are also some kits that enables you to do like a real-time real-time PCR. So they tell you also more precisely which bacteria you have, but also again, they can be not alive.
The most, in my opinion, the most risky microorganisms are fungi, not bacteria.
But depends from which side you look. I was looking.
We can also try to find this type of kits to be able to detect the presence.
Fungi, I don't know if there are kits. You can create it yourself with time and money. I know that some company also send like kits which you can customize, but I don't know if they have the assay for fungi. So I always look at it from the human point of view. So it was like to preserve the health of the workers. From the microbiology point of view,
It's more difficult.
European project.
European project. Okay. Thank you. Thank you very much,
You're welcome.
Lisa.